Everyone. So my name is Duke Mu. So I'm currently a 40-year graduate student and working in Dr. So's lab. So today I will talk about the Discover 2025 poster, which is the roles of UN's nurse-specific protein in the post-transcriptional modification of the pre-mRNA. So first, I will talk about the background, which is about the splicing. So pre-mRNA, once they go through transcribe, they need to go through the splicing, which is initiated by small nuclear ribonuclear protein. So here we have US nerve recognize the fragment splice site, which is the junction between the exon and the intron. So when they recognize these, they can initiate splicing. So more of my focus will focus on the US nerve. So U1 nerve, they have additional function named telescripting, where they prevent premature cleavage and polyadenylation when the pre-mRNA gain transcribed, so they can produce full-length pre-mRNA. So U1 nerve contain three specific binding protein, which is U170K by at stem loop 1, U1A by at stem loop 2, and U1C by at stem loop by at at the fiber splice site. So US nerve also contain the SM protein where they can define the maturation of the US nerve. So they can get these SM protein when the US nerve undergoes its own biogenesis. So when U1 snRNA get exported out into the cytoplasm, they can get the SM protein by the SMN German complexes. Once they get the SM protein, they can get back into the nucleus to do splicing. So my study will more focus on U1C to see how U1C can associate with U1 SNRP during its own biogenesis. So we developed the in vitro reconstitution of U1 SNRP by combine the U1 snRNA with the recombinant protein and SM protein, and then look at the Western plot with the immunoprecipitation so we can see if U1C can associate with the U1 SNRP. So when UNC, which contains several basic residue at the N terminus, when we mutate the basic residue, UNC is losing its own binding capability to the UNC. So we learned that UNC basic residues are important for the association with UNC as well as the UNC. So UNC also may associate with the SMN and German complexes throughout these basic residues as well. Another project we have been recently initiated is the liquid-liquid phase separation of UNC protein. So UNC in the C terminus contains several arginine protein residue which can promote the liquid-liquid phase separation when mixed with the PEG 8000. And then we can see the droplet formation under the confocal microscopy. To have more clear visualize, we will also incorporate with the fluorescent tag to see any droplet formation under the confocal microscopy. And finally, another project we are working on is the dual luciferase reporting system, where we can utilize the 2-exon 1-intron system with the firefly at the N terminus and the granular luciferase at the C terminus. So when we incubate the US nerve together, they can clone and then we can record the ratio between the granular and the firefly. So we can monitor in real time for the splicing as well as telescripting. So from the conclusion of my study, that UNC basic residue can be used is required for the association with UNC. And then the droplet formation also can be used to study the association of UNC with the SMN German complexes. And finally, to monitor splicing and telescripting, we can utilize the dual luciferase report system, which is currently we are working on. And then finally, with the acknowledgement, we want to thank our collaborator as well as UTA for this opportunity for my presentation. Thank you.